First of all, as the slides sort of switch over, thank you very much. I'm not an archaeologist, but I work with archaeologists, so I do get to do a little bit of, the passion that I had when I looked at, different documentaries and, wanted to be an archaeologist, but I couldn't really get to be an archaeologist, but I still get to excavate. I just excavate DNA sequences rather than actually excavating, human remains or other cultural artifacts and material artifacts from archaeological contexts. So let's go ahead and set the scene here. So I really hope that in the next hour or so I can give you a brief appreciation. It's a short time. There's a lot of material to go through, so I've been fairly selective. It's bits and pieces, snippets of work that have been published. But of course, in terms of the motivation for this particular session, as Uma, already mentioned, this has been a really exciting year for us working, those of us who work in ancient DNA, but also generally in evolution and evolutionary genomics. It's the first time that the Nobel Committee has decided to acknowledge the actual application of sort of basic science, because that's what evolution comes under, in sort of the broader context of medicine and the physiology, of course, award that went to Svante Pääbo. He is, as Uma mentioned, he's probably the intellectual forefather for many of us who are working in the field today, because he, set up many of these methods and sort of even just the chance to take the chance of starting on a new field and pushing the limits of what's possible to answer. So he, along with multiple other people, all of this work started in the University of California, Berkeley. So the first paper that actually, managed to sequence DNA from something ancient, something extinct, was published in 1984. So this was not from humans, it was from an animal. This was an extinct animal called the quagga. It's closely related to modern-day zebras. And that was from a little piece of muscle that was hanging from the last living individual who was then put on display in a museum in South Africa. So that was sort of the start of the field. This was pre sort of the big, revolutionary technologies that we have today in our possession. So just thinking back to what little they had, and yet, to think and to envision and to think big and really, go out there and take all sorts of risk, and in the process really kind of invent a new field. So really thankful to all of those folks who dreamed big, because that gave me a job today, right? So I think taking a page from Dr. Pääbo's achievement this year, of course, he won the Nobel Prize for, cracking the mystery of what makes us human, how did we evolve, and that's to the sequencing of these archaics, and we'll meet them in a few slides. But before we go there, I guess I just wanted to situate ourselves in this story. So the story of humans starts in Africa. There have been multiple episodes of different forms, let's say, of humans and ancestral, species that have originated in Africa, who've left Africa and made it out multiple events, multiple times. And so this is sort of the characters. There's many of them. This is just one slide that I took from the Natural History Museum in London, their website. These keep changing as more finds become available from the paleoanthropological record. You can see that this particular timeline stretches to about 7 million years. And they've sort of tried to kind of situate early hominins and various sort of forms that come over the past 7 or 8 million years. And within that, you'll see sort of the bigger human family, so those who are more closely related to us. But if you actually see Homo sapiens, anatomically modern humans, that's us, you can see what a tiny blip of that history we are, in fact. So our history is just the last 70 to about 100 to 200,000 years. So in the grand scheme of things, we're yet a tiny blip in the history. I just put here for context, so the chimps are our closest sort of primate relative, and they sort of they already kind of diverged from our lineage about 7-8 million years ago. So that's sort of the genetic distance, the temporal distance of the DNA of the chimps to us. And then you have all these early forms, and some of them are displayed here. So we have, I believe, we have the Australopithecus afarensis, if some of you may have heard of the famous Lucy, the fossil of Lucy. So that's that species there. And I think I don't think it's Lucy, but it's some other Lucy's relative there that belongs to the same species. We also have the Homo erectus sitting there, so that's within the human sort of family. We also have the Neanderthals, and we'll meet the Neanderthals a bit more in my slides. And then we have, of course, Homo sapiens, and we have the chimp skull sitting there. So after the talk, please feel free to come and engage. So we'll mostly focus kind of on the recent history, because that's really what bears upon our DNA and sort of what we are as humans. But I just wanted to introduce you to this broader family of relatives that we've had. Most of them have lived and developed, evolved in Africa. And then we've had sort of this exodus, of course, of our immediate ancestors who came out, and hence we're all now globally, global citizens, anatomically modern humans. So there have been a lot of debates, some of them still persist, in terms of what are the models of the origins of our species, of anatomically modern humans. So there's been a range of models and range of kind of scenarios that different paleoanthropologists or anthropologists have proposed based on various data, but also sometimes just out of intuition, let's say. So these are the two kind of ends of the spectrum of the models that exist or existed in the literature. So on the left, we have what's called the multi-regional model. So multi-regional model posits that you have basically one common ancestor that lived about 2 million years ago, potentially Homo erectus, came out of Africa and sort of went and settled different continents. So here you have different continents. And essentially, since that 2 million year exodus, they just settled and lived kind of isolated in these different continents, right? So it's kind of this continental model. But however, given that today, all of us, of course, we all humans live in different continents, but we're able to interbreed, we're the same species. So to justify the fact that we are still the same species, this model kind of allows for a little bit of genetic mixing so we don't drift and become different species over time. But essentially, this is sort of more of a continental settlement, and then you sort of just each continental kind of population evolves on its own, right, with some contact between those. On the other end of the sort of spectrum, you have the more recent out of Africa model. So, and then this is sort of the total replacement sub-aspect of the model. So what does this posit? So this still says that there is an out of Africa, because you find erectus outside as well. So you do have the 2 million year old erectus coming out. At some point, it forms these other archaic, so you do have Neanderthals, Denisovans, possibly other forms that existed. And then on the other hand, you have modern humans, or the lineage that forms the modern humans. However, it's not that they sort of settled, unlike this model, that they settled different continents and then that was it. What this says is there was a second migration more recently out of Africa. And what this suggests is that everybody who lives out of Africa today, so all of us non-Africans, we come from this one more recent migration out of Africa, which happened about 60 to 100,000 years ago. So the difference between this and this, both of them say that all of us have we have African origins, but it's the temporality that's different. This one suggests a much deeper sort of temporality, right? It's about 2 million years, whereas this one suggests that we have more recent roots, those of us who migrated out of Africa. So without going into too many sort of technical details, I'll already give you the spoiler here. So, so far, most of the genetic data suggests that it's the out of Africa model, so the more recent migration that's the most likely model for the origins of us, anatomically modern humans. And that's simply because if you take our DNA, DNA of people living in different parts of the world, and you look at when was the last time we all shared a common ancestor based on our DNA sequences, that's much more recent. It doesn't go back to 2 million years ago. We're connected more recently, and that more recently actually matches up well with this. So in the last 100 to 200,000 years. So it very, very neatly supports the recent out of Africa model. But keep in mind that what this says, the second aspect of this model is a total replacement. So what the total replacement part means is that when our ancestors came out of Africa, let's say 100,000-ish years ago, we replaced all the other forms of humans who lived there. So Neanderthals, Denisovans, other forms that existed, possibly erectus. And so there was this idea that, we don't know how. It could be more, people have suggested violence, that our ancestors came out, wiped everyone else away. There's also been a theory of, disease exposure, so that our ancestors came out, brought all sorts of diseases that, pathogenic variants from Africa that maybe our ancestors were living with, so they were adapted to. But then these naive populations of other hominins who lived outside of Africa were not able to cope, so they got infected, and that wiped them away. And there's many other such theories. So far, we don't have conclusive data to actually, confidently ascertain which of these theories is correct. Why did these get wiped off? But also, I want to revisit this theory, so do keep this in mind, that we do have evidence for out of Africa, but this total replacement part we'll revisit in a few slides again. So just keep this in the back of your mind, it'll come back. So in all of this, you still have an exodus out of Africa. We start in Africa, we come out of Africa. Of course, we all, many of us live outside of Africa. So there's this concept called the serial founder effect. I'm not going to throw too many jargons, but this will be one of the few, but I'll also break it down. So the serial founder effect is this idea that, you have this massive, the original kind of genetic diversity is in Africa because that's where we originated. So that's why the entire pool of the DNA diversity exists. And then, 100,000 years ago, when that out of Africa migration happens, you have this what's called a genetic bottleneck. And what that essentially means is that a random subset of the ancestors will leave, right? So it's kind of like, you have a bag of M&Ms, you put your hand in, you take some M&Ms out. You may be able to capture all the colors, or every random draw, you may get some colors left out. That's not in your control. It's just a randomness of, the stochasticity of the process. So very similarly, it's the stochastic process that may lead to, let's say, loss of a few these colors representing the genetic variation, right? So for example, let's see, what do we lose here? We lose the yellows. So the yellows are gone. You only see them now in Africa. And then you get further, movement from, let's say, the Middle East as people come into, let's say, South Asia. You're losing more colors. You lose, I don't even know what you lose here, but some color, maybe the reds or something. Red. Okay, thank you. And so on. Here you lose the blues. So the whole point is that as you're moving away, your genetic diversity gets bottlenecked, right? And so one of the things you want to see if the out of Africa migration theory is true is that the further you get from Africa, you should be seeing or recording lower and lower amounts of diversity in populations further away. And that's exactly what we see. So this is empirical data. This was already established back in 2005. And this is data from living people. So we are not getting into ancient DNA yet. So this is living people who were sampled, whose DNA was sequenced and analyzed. And so the way to read this is on the x-axis, you have distance from Africa. So if you go from left to right, you're moving further away from Africa, right? And on the y-axis, this is the genetic diversity. It's just a proxy for how diverse or how much genetic diversity there is in these populations. The different colors represent different regions of the world from where the individuals who were sampled came from. And so you'll see in black are African populations. Obviously, they'll be closer to zero because you're measuring the distance from a central location in Africa. So they have the highest numerical value on this on this y-axis, which means they have the highest genetic diversity. You come a little bit further, you see Europeans in blue, then you see the Asians, they're a little bit lower, so you see this trending downwards. Then by the time you get to Oceania and finally the pinks that are the Native American populations, you've got this downward trend. And this squares really well with, of course, the increasing distance here, right? So this also sort of was taken to kind of interpret the route or the way that the world was peopled. So just by looking at this, what you can say is that perhaps the populations who ended up in the Americas, who are the ancestors of the Native Americans, were likely to be some of the last kind of that sampling, the subsampling as it happened, right? And of course, this also is, we should take a little bit of, caveat here, which is that this was back in 2005. We're still struggling with an underrepresentation in terms of how many populations in the world have been sequenced. So that was even less so in 2005. So perhaps if you sequence, some of the island population in Polynesia or something, they may be probably similar or lower. So this is sort of missing a lot of regions. So this is very, very sort of broad continental scale. But this is sort of the general pattern. So this was kind of, also one of the reasons why sort of the out of Africa model was supported. This was one of the kind of main nails in the coffin against the other model. So keep that in mind as we move on. So over time, though, we started, that was data that I showed you from 2005. And now we're in 2022. There's a lot that has changed. So we've been very fortunate to have a lot of amazing technologies come our way, generally, globally, but definitely in biology and molecular genetics. There's been a lot of technical development. And that has led to, or that's opened up avenues that we never thought would be possible, right? In fact, and this is a little bit of an aside, but just last week, there was a record-breaking paper that sequenced DNA that's 2 million years old from Greenland. Never, I think, when I was a PhD student, in fact, the author of that paper is my PhD supervisor. Even being mentored in that lab, I don't think we ever thought that anybody could break, the million-year barrier. But here we are, sitting at 2 million years old. And so a lot has really improved, which also means that even for the study of our own species, we've gained a lot of information since those early days. Of course, those are seminal studies upon which a lot of these later studies are based on. But we have now moved to a space where we've got so much data, we've got so much information that the models are becoming more and more refined. On the other hand, we also have, as more data comes our way, there's also many, many unanswered or unresolved aspects. So there's that challenge in science that sometimes more can also be a little confusing, or you'd always want a little bit more to be able to then come back and answer some of these or put the whole model in perspective. So this is a figure from 2017. It's already become outdated in the last few years as more data has come out. But this is basically based on new data that came out since 2005, particularly in the form of DNA from ancient or extinct individuals, ancient DNA. And so there's two aspects of this figure that I really want to highlight here. So first of all, just to orient you, this is timeline going from present day, so modern times, all the way to ancient times. So we're at about 650,000 years here. And so this is supposed to cover the entire sort of timeline of the sort of evolution of our lineage here. And so you're here in Africa in the left, right? So you have sort of here divergence of our kind of lineages from the Australopithecines and all of that. So you have the formation of sort of some lineage that's closely related to our ancestors, which sort of moves in time. And somewhere between 150 to 200,000, we have the origins of modern humans, as in the way that we look anatomically modern humans, so our direct ancestors in Africa. So all of this, we're still in Africa here, right? And then about 100,000 years, you start seeing the sort of diversification of our own ancestral lineages. So this is something that we call in population genetics, we call it population structure. So basically means the population is not homogeneous. There's also differences within the population, genetically speaking. So you have these subpopulations. And so you have this early diversification already happening in Africa. So when you have the out of Africa migration, the one that I spoke about, the recent out of Africa model, so that's depicted here by this little thing here. By the time the early structure in Africa is already set up. So we don't have like one big pool from which ancestors come, but actually multiple sub-pools, and any of those pools could have contributed to all of us non-African diversity. So we still don't know much about Africa because a lot of ancient DNA actually comes from outside of Africa, and the main reason for that is that ancient DNA, as you can imagine, these bones have been buried in the soil for, in this case, hundreds of thousands of years, and it simply will not survive, particularly in tropical regions and conditions in Africa where it's hot, humid. And South Asia actually falls in that sort of suboptimal environment as well. So it's very, very challenging to work on ancient samples from places like South Asia, South America, Africa. And particularly when you go into these very, very old timelines, there's absolutely no way that most of these fossils will have much DNA. So we're missing a huge chunk of our understanding of who exists, who are the actual ancestors from where this out of Africa migration happened. But let's see, never say never. We couldn't guess the 2 million barrier, but that happens. So who knows, at some point, if we get lucky, get even that one sample that survives and preserves well, we may be able to actually, get some information about the ancestral population. So moving on here. So here, actually, I neglected to tell you, of course, when this diversification is happening, that's kind of when you have these different hominins, that we see out of Africa as well, the Neanderthals, Denisovans. In fact, these people also put unknown hominins, which is, I think, it's a good thing to do because, we're never going to have the full picture. There's going to be all these unknowns, many of them that paleoanthropologists have already dug up, that we don't have DNA from, but also things that maybe paleoanthropologists haven't yet dug up, and particularly in Africa, where, there's a lot to be done in terms of archaeology and paleoanthropology. And so there's all this diversity that exists, and then of course this particular lineage that's then giving rise to all the modern non-African populations here that are all basically a subset of the diversity in Africa, which we just saw. So this is sort of the map. This is also supported by ancient DNA, but I'll give you two main layers of information that have anchored this particular figure that was published in 2017 that ancient DNA is added to that wasn't particularly known before because we didn't have access to this data from the past. So one is actually the identity of these archaics, right? So before the Swante Pabo's team actually published the genome sequences of Neanderthals, we knew them for what they were, a skull, pieces of bone. So, of course, paleoanthropologists had this identity established. And it was possible to do that because morphologically the Neanderthals do look different. I'm not going to say much about that because it'll be more exciting to have you go and compare the modern human skull with the Neanderthals and actually see that you can tell them apart. There are very striking features that you can use, which is what paleoanthropologists use as well to tell apart different species. But ancient DNA, of course, gave us another identity of these individuals, which is the genetic identity. And suddenly we could then compare the DNA of these very closely related humans to ourselves and actually try to ask a question which Swante himself asked: what makes us human? What makes us unique? Right? Why are we different? How is it that we have survived? Some of these guys have died out. So all of these questions now become more accessible because we have more information than just the morphology of these individuals and of these different archaics. The other large piece of puzzle that has been sort of, we've moved closer to broadening our understanding about is ourselves. So in addition to the archaics, also just understanding the differences and similarities of us modern humans as we exist across this globe. How did we spread across the world? How are we all related to one another? How did all these evolutionary processes pan out in the last 100,000 years as we moved out of Africa? And how does that make us different from the ancestral populations living in Africa? So all of these questions. Now you may ask, well, why can't you answer that already? We're all living, we can sequence each other's DNA and just compare and talk about evolution. Sure, and that's what was done before ancient DNA became a big thing. But there's a lot of challenges in understanding and just studying people who live today. For one, if we just study people who live today, we would not have known the genetic identity of these extinct hominins. And much like the extinct archaic species, there's also ancient DNA is bringing to light the fact that there's also many anatomically modern human lineages that have gone extinct or are very rare today. So no matter how many billions of people we sequence, there may still be parts or pieces of our history, of our prehistory, that we may not be able to capture just by sequencing people who are alive today. The second challenge is that, and something that I'll talk about a little bit more towards the end of my talk, is that all of us living today, we are a big genetic mess. We are all mosaics of one another. And this is something, I think, just philosophically and romantically speaking, is just such a beautiful thing that we think about all these barriers that we've made for ourselves. But actually, when you look at our ancestors, they were mixing freely. And how freely, and I'll show you some examples of that. And so the problem then becomes that if we are mixed to large extents, that's going to sort of almost make it harder to see distinct kind of genetic origins, because there's more that we all share than are different amongst us. So this mixing has, on the one hand, brought us all genetically closer, but on the other hand, it makes it harder for us, particularly in human evolution, to actually piece apart, let's say, the distinct genetic histories in many cases. So we may still be able to do a good job, but not to the extent that adding this ancient DNA layer would actually help us refine these models. And so that sort of brings me to our player here. This is, I thought this is a really nice cartoon that captures the kind of work we do. The sort of collaborative effort. So there's no way that somebody like me can just start a lab and work on ancient DNA. We need our social science colleagues, particularly anthropologists and archaeologists, to actually work closely with us and identify which samples, what questions to ask, what the context of these samples are, so we can actually think about devising hypotheses and actually sequence the DNA and also be able to interpret that in a holistic fashion that may not be possible just by geneticists, because we're missing the huge context and the social science perspective. So it's a highly, highly collaborative, multidisciplinary undertaking that we do. Very briefly, I'm not going to, again, get too much into techniques, so we can take this maybe in Q&A, just to save some time here. But a lot of people often wonder where ancient DNA comes from. So from the perspective of us humans, most of the ancient DNA papers have been able to use bones and teeth. Those are really the parts of the skeleton that survive long in the soil. There are a few studies that have also made use of hair. It's not as rich a source of DNA as sort of directly your bones and your teeth would be. So those are really the most common tissue types that come to our labs when we do DNA extractions. In addition, more recently, there's been creative uses of other sorts of inputs. So, for example, this is a little sediment core, ice cores, and the 2-million-year-old DNA record breaker was an ice core that was taken in Greenland. Now, obviously, that's not going to give us genome-level data from the individual, but these things can be used to, let's say, reconstruct past environments by looking at what sort of DNA from different insects or plants or whatever gets trapped in these sediments. There have also been cases, in fact, Svante's team, the team from Leipzig, has used sediments from cave sites across Europe Cave sites are particularly good because you can think about these as habitation sites, shelters in the past for our hunter-gatherer ancestors or Neanderthals. And so they've actually been able to use these non-conventional input types to be able to actually even detect tiny traces of Neanderthal DNA to be able to say that there were Neanderthals here at this time. And this often becomes important when you don't have fossil evidence. And fossil evidence may not always survive, or maybe it hasn't been excavated. So just to even broaden our understanding of who lived where when, some of these new ways of thinking about where we can get DNA from the environment are actually becoming more popular. So I just wanted to give you a sense of what we work with in the labs, right? So I mentioned to you that the last sort of 10, 15 years have been especially seminal because of the techniques and the technologies that have made it possible for us to actually gain access to this really very, very useful, almost as Uma said, a time capsule, right, ancient DNA. So two of the main sort of improvements in methodologies and techniques have been that we as a field have been able to identify which parts of the skeleton, particularly in the case of course, those of us who study modern humans, preserve DNA better and for a longer time. So different parts of your body will have different, bones of different density, of different porosity, and so that will dictate what encapsulates DNA best and also protects it from environmental degradation. So often when your bones are buried in the soil, it'll get acted upon by the bacteria, the soil microbes. You'll have things like humidity in the soil or the temperature, the microenvironments. All of that will dictate how well the DNA survives. And so your long bones, for example, right? So all of these, I don't know how many of you have had a chance to look at them, but they're all kind of porous. So there's a lot of tiny, 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 tiny channels, and that's sort of the intrusion that allows bacteria or free water or whatever to seep through, and that causes havoc with the cells and sort of the DNA that's encapsulated inside the cells. So that being said, there's two particular types of bones, or teeth included, that actually protect DNA longer term. Of course, one is teeth, particularly if they're intact, the roots are intact. Most of us become part-time dentists in our lab, so we actually go in with dentist drills, cut this, go in, get the pulp chamber out, and then that gives us a fairly decent source of DNA that's protected for the most part from the soil microbes. So we get more human enrichment there, right, organically. The other bone that's been, particularly in the last 6 or 7 years, that's come to light in terms of preserving DNA even better, often compared to teeth, is what's called a petrous bone. So this is, petrous means stone-like. So this is the bone that's sort of inside your inner ear. So you have the ear canal, so it's the bone that surrounds that, and of course you have two. And this is one of the hardest bones in your body. And by virtue of being hard, that means it doesn't have porosity there, so it doesn't let any environmental factors kind of intrude and ruin the DNA integrity. So those of us who go around collaborating with archaeologists or museum curators, we often tend to ask for these because it gives us a fighting chance against DNA degradation. The other big, of course, technological innovation which has come as almost godsend for the entire field, in fact, not just ancient DNA, but also anyone who works on anything that requires DNA sequencing, is these big, what are called the next generation sequencing machines. So these are these massively parallel high-throughput machines that churn out, millions and billions, in fact, of bases of DNA in a matter of a few hours. So to put things into perspective, the first human genome that was sequenced, it took 10 years to put together the first draft of the human genome, right? That was the early days. It required a lot of manual effort and international consortia, like tons of researchers who had to put in efforts. Contrast that with today. I can walk up to one of you, ask you to spit in a tube, go to my lab, extract DNA in a couple of hours, send it to a company. By tomorrow or day after, you can get your data back, right? And really good quality data too. So that's sort of the improvements in technology, which also means that it gives us, of course, quicker throughput, quicker access to data, but also compared to older technologies, the best thing about these technologies is that it requires less input. And this is, of course, very, very important for those of us working with culturally important heritage. These are biocultural heritage for a lot of people, their ancestors. So we can't go around drilling big holes in them. So we, these days, require very tiny bits. Our methods have improved in the lab as well. So a lot of these, although they are destructive sampling, but they're minimally destructive sampling. So we don't have to upset museum curators or sample custodians, or in many cases, the communities who hold, who revere their ancestors, of course. So just to give you quick glimpses, so we work in ancient DNA labs, and the reason why you don't find an ancient DNA lab in every nook and corner is because these are very expensive forensics grade labs. So as I told you, because ancient DNA degrades very rapidly, if we analyze it in a lab that does, let's say, modern DNA analysis, there's a huge chance that we may cross-contaminate, and our samples will be flooded with DNA that somebody else is extracting from, let's say, a living individual. And so to be able to convince ourselves that our data is good, it's solid, and also, of course, convince the scientific community and the readers at large, all of us work under very strict, very sterile conditions. So these are facilities that are able to only process what we call low template degraded samples. So for example, this is a glimpse of my lab at the University of Chicago. Here's my student working. You can see fully PPE'd. So head to toe, the only thing that's visible is your eyes. And again, the reason for that is that we are, most times we are the worst enemies of our own samples. If you're not careful, we can contaminate our samples, and then you'll end up sequencing yourself and not the ancient individual, which I'm sure is not something you want to do. So very common things we do, for example, it's positive pressure. So this clean air that gets pushed through HEPA filters, and the dirty air gets taken out, and this air change happens multiple times in an hour to make sure the air is very, very clean. Every person works in their own space, so no two people are sitting side by side, because even if two people are working on ancient samples, they can still cross-contaminate each other's work very quickly. We bleach every surface. There's UV lights all around. I don't know, you probably can't see it in this photo, but we have UV lamps all over the ceiling that come on between, let's say, midnight and 6 a.m. that just zap the entire space overnight, and then it gives us sort of a fresh working environment the following day. So consequently, it's very expensive, right? You have all these air changes, you have the UV lights burning through the night. So not a lot of institutions are able to afford the upkeep, because there's one thing about building infrastructure, but there's also the second thing is the maintenance of it. So not a lot of countries have the facility, or even if they do, it'll be one, right? And that may or may not be sufficient. But just to kind of put that out there, that there are challenges in terms of finance, in terms of infrastructure that a lot of us face. So all of that being said, let's jump into sort of the story for which you're here. So of course, in Leipzig, in Svante Pääbo's lab, they have a very impressive facility, probably more impressive than the one I showed here in my lab. And that's where all of the seminal work that eventually led to the Nobel Prize this year was done over multiple decades, huge teams working. And so I'll talk, the rest of my talk will be focused on sort of these archaics and really trying to understand their genetic relationship to us. And then I'll revisit that total replacement out of Africa model, and maybe you can tell me whether it's right or wrong. And that'll tell me how correct I was. 10 minutes. I only have 10 minutes. Oh, no. Okay. All right, that means I need to speed up. So this was the cave site from which the DNA sequences that were published from Neanderthals came. It's a cave site in Croatia. These are some of the samples. And so, of course, they sequenced the DNA of this Neanderthal in Germany, in Svante Pääbo's lab. And what they asked was, when you look at the DNA sequence, this is a random sequence here, but if you compare the DNA of a Neanderthal to multiple, any pairs of, let's say, modern humans across the world, is the Neanderthal sort of, closer to one or the other? Now, if the Neanderthal was sort of, what we call an outgroup, or somebody who's ancestrally related to all populations, then the Neanderthal DNA should not be closer to one particular population versus the other. And so when they compared two, let's say, African populations, that's what they found. They didn't see that the Neanderthal was closer to one African versus the other. So it was equally distant, what we call an outgroup. Okay? And so they repeated this with other pairs of populations. And what they quickly found was when one of their populations was African, and when the other populations cycle through non-African populations, so let's say African versus European, African versus Asians, and African versus Oceanians, pretty much all of the non-African populations were significantly closer to the Neanderthal sort of sequence, right? And there's very few scenarios where this is possible, at least knowing the total, the out of Africa model. And that would be that if there's been sort of interactions between this Neanderthals and non-African populations. So we'll come back to this model again, but I quickly want to just introduce you to the second character in the story. So the same year, in fact, when the Neanderthal draft genome was published, 2010, the same team, Swante's team, along with a number of researchers, including David Reich from Boston and multiple other researchers, published the DNA from yet another hominin. So they called it the Denisovan based on the name of the cave, Denisova Cave in Siberia, where it was excavated. Prior to this, there was no fossil, nothing. Nobody named this as such, which is why they named it after the cave. And the reason for that is the only piece of bone that actually identified this particular, archaic was this pinky bone. So you can see this teeny tiny piece that actually was used to extract the DNA sequence it. There's no way one can, the most expert of experts can look at this and say, oh, this is a new species. Maybe somebody can, but until then they hadn't. So this was a completely new find based simply on protein and DNA sequencing. So again, this shows the sort of power or the complementarity of ancient DNA, where it can add to detail that may not simply be possible by looking at a piece of bone that may be overly fragmented, even for experts. And so this particular, when they analyzed the DNA, they basically saw that these Denisovans were very close to basically all the Neanderthals who had been sequenced. So that was sort of their genetic distance, and both the Neanderthals and Denisovans are sort of, again, what we call an outgroup to all the modern humans, the anatomically modern humans. So these are Africans, East Asians, Oceanians, Europeans, another African population. However, when they repeated a similar analysis as what I showed you, so when they sequenced the Denisovan DNA, and again asked between pairs of populations of modern human origins, what they found was when they, let's say, compared African and African or European and Asian, for the most part, Denisovan was equally distant, so it wasn't any closer to any of these populations. However, when they brought in Oceanian populations, so populations from Papua New Guinea and sort of the island nations there, what they found was this particular pinky bone individual was a lot closer to Oceanian populations than to any of the other populations of the world. So that was fairly exciting. And let's skip this and go to the other slide. So this sort of forms the basis, the model here. So here we have the African modern human populations, we have Europeans, we have the Han, we have the Melanesians populations, and then we have this sort of scenario now based on ancient DNA data, where you have the Neanderthals who contribute to basically all non-Africans, as we just saw, right? So this arrow doesn't go into the African part, just to all the non-Africans here. On the other hand, we have the Denisovan, for whom we have absolutely till date, we don't have a fossil in the way that we have Neanderthals, only recognized by that teeny tiny pinky finger piece of bone and the DNA, of course, now. So this particular hominin contributing to Oceanian Melanesian populations only for the most part. More recently, it's been shown that they also contribute a little bit to East Asian populations, but for the scope of this talk, I'm just going to say that primarily the contributions were to the Oceanian part of the world. And so this is sort of where we come into this concept of archaic introgression. So archaic being these forms, and introgression is another word for sort of DNA that comes in from these distant to, of course, our anatomically modern humans. So then we come here. So we're back to these, if you remember, the start of my talk. So we'd already excluded the multi-regional model based on actually DNA, for modern living individuals. We said that we had a scenario where the recent out of Africa model was true. But we mentioned in this model, it posits that there's total replacement, that humans come out of Africa and they sort of replace the Neanderthals and Denisovans. So that being said, and in light of my last few slides, who believes that this model still holds true? Raise your hand. Yeah? No one believes this is true. So we should discard this in favor of another model that does what? How would you change this to reflect what I just showed you? There's some hand waving going on there. Anybody? Just yell it out. Right. Absolutely. So we'll cross that out. And now we have the introgression model, where you basically have, very simplistically, these arrows going in. So it's also called the leaky hypothesis. People have different names. But really, the point is that while we all have very recent origins, we weren't isolated. Not only did we sort of intermix amongst ourselves, we also heavily intermixed with other hominin forms that were alive and kicking around back then. So very quickly, just contextualizing this. Now, it's important to note that whenever you see maps like this, very often, don't take these to the T. There's no way DNA can tell you exactly which path anybody walked. So these are all supposed to be kind of more broad-scale movements across the globe. So the current sort of understanding is that once humans left Africa, again, this happened sort of 60 to 100,000 years ago based on archaeological evidence. Sometimes between 50 and 60,000 are, the non-African ancestors meet with Neanderthals who are hanging around somewhere in the Middle East. Again, we don't know exactly where. And then, boom, this first introgression happens. So basically, this leads to all non-Africans, depending, wherever you are. It could be the Americas, it could be Europe. All of us have a bit of Neanderthal ancestry that Africans don't have. Or at least most Africans don't have. Some of them have it because more recently there's been backflow, but anyway. And then a few sort of 1,000 years later, you have the second sort of introgression pattern that happens. Again, we don't know where, because clearly the Denisovans are up here, but the Oceanians today are living down here. So it may happen anywhere in this territory. We don't know yet. We need more ancient DNA evidence. We need more fossil evidence. But clearly, there's been a second introgression that's restricted today to Oceania that involves a second player here. So this is sort of just to put the visual in terms of where who is at which given point of time. And in terms of how much contributions we have, so this is a map that shows, it's sort of a heat map that shows the percent or the proportion of Neanderthal ancestry in different worldwide populations. So each circle is a different population of the world. And the higher or the warmer the colors, that means higher the ancestry. So technically, everybody that's non-African has some level of ancestry from Neanderthals. There's a bit of higher, signal here in East Asia, and I won't have time to talk about it, but if anyone's intrigued, we can chat a bit later. But generally, on average, between 1% to 4%, normally we say about 2% to 3% is what most of us have from Neanderthals. And if you do a 23andMe, this will probably pop up around the same percent for most of us, right? And similarly, for Denisovans, I'll just skip ahead and then come back to this. So this is the Denisovan landscape. So again, we have very, very high percent here in Oceania, as I mentioned, up to about 5% in the Oceanian genomes, they have Denisovan ancestry. So one thing you may ask is, how do we, I said about 50 to 60,000 years ago is when the Neanderthal sort of introgression may have happened. So of course, there's sort of intuition there based on fossil evidence, but you can also use DNA sequences to date So we have a molecular, dating. It's a molecular clock that can tell us when certain events have happened. It's not overly precise because there are assumptions to be made, but it does give you some level of a ballpark of when certain, introgression or admixture events happened. So in this particular case, I want to introduce you to this player called Ust-Ishim. So Ust-Ishim is a site in Russia, in Siberia. It's right here. And here, a 45,000-year-old individual was found. Their morphology was similar to anatomically modern humans compared to Neanderthals. So they looked more like us than Neanderthals in terms of their fossil morphology. But they're intriguing because they're very close in time to when humans moved out of Africa. And so they've, again, Svante Pääbo's team sequenced their DNA. And among many, many cool things, what they found was this. So this particular map has Ust-Ishim on top and different modern populations from different parts of the world. And all I want you to take away from this is that these sort of marks here, these yellow marks, these kind of track the length of Neanderthal sort of related DNA that's in these particular populations, right? In this case, these are the tracks of Neanderthal that are in Ust-Ishim. These are the tracks in French. So immediately what becomes clear here is that the Ust-Ishim, that ancient individual, has longer tracks of Neanderthal. And what this really translates to is the longer that you have tracks from the donor population or the source, that means you're closer in time to that event. Because we have something called recombination in our body, and recombination is a process that breaks down your DNA chunks over time. So, which is why all of us modern individuals have shorter chunks, because it's been much, we live today, and that events happened in the past. Whereas Ust-Ishim lived 45,000 years ago, and they lived much closer in time to that mixing event. And so that mixing event has to be sometime before 45,000 because this Ust-Ishim already has the Neanderthal ancestry. And based on the length of the fragment, which is about almost 1.8, almost 2 to 4 times longer in Ust-Ishim compared to present-day individuals, you can use sort of an algorithm or a calculation to date when that admixture happened, and based on the molecular dating from the DNA from this ancient individual, we can say it's happened sometime 50 to 60,000 years ago. So that's sort of the current understanding of when our ancestors who left Africa met and interbred with the Neanderthals somewhere in the Near East. How am I doing for time, Mama? At time, probably. Okay. So very quickly then, I'll also give you, a bit more of a functional sort of aspect to this. So, which is also something that has motivated Svante Pääbo's team to look at a lot of the biological consequence. So it's one thing to say, well, we have ancestry from Neanderthals, Denisovans, but what is this foreign DNA doing inside of us, right? Because if you think about it, our bodies shouldn't be tolerating foreign DNA. It's intrusive. Yet we retain segments of DNA from these, other archaic forms. So it must be doing something. And this is still early days, so we're still investigating. People are doing functional analyses, various sorts of analyses to understand the pieces of Neanderthal and Denisovan DNA that are still hanging out in our genomes. Is it doing anything? If so, is it good or is it bad? And so a very quick example of the good that it's doing. So this is an example from Denisovans. So as you all know, Tibetans, they live in high altitude. It's a hypoxic environment, which means it's low in oxygen. Yet these Tibetans are well adapted. Whereas people like you and I, if we go, we need to acclimatize, right? Many of you may have heard, like if you go to Machu Picchu or something in the Andes, you need to go a few days ahead of time, or even to Ladakh, right? They ask you to come a couple days, get used to the hypoxia, then do whatever activities you want to do. And so people, particularly this team from Berkeley, they started analyzing the available data from living Tibetans and lowland Chinese populations. And by 2016, the Denisovan genome was available to analyze. And so they focused on this particular gene called the EPAS1. So this is a particular genetic marker in our bodies. And this particular gene controls our response to hypoxic conditions, right? So this is what gets activated and what activates the chain of hypoxia-related events. And this is particularly, it's been already known even before the study that this obviously, in Tibetans, this has some sort of a kick to it. So it does come up in natural selection studies. But it was still not known how come only the Tibetans have this, right? When did they get it? Because they are closely related generally to the lowland Chinese populations, yet they have this really strong signal in this particular EPAS1 gene. So when did they gain that? And so when they compare the sequence of the EPAS of the Tibetans to the Han Chinese, to other global populations, and to the Denisovans and Neanderthals, what they found that the only other individual that had an identical sequence to the Tibetan motif was the Denisovan genome. And so this is a classic case and probably the most conclusive of cases that we have so far of what we call adaptive introgression. So it's introgression coming from the Denisovans, but it's adaptive because it's given the Tibetans the strategy to survive in an extreme environment that most of us other people can't because we don't have that particular mutation or the sets of mutations that allows them to survive in hypoxic environments. So this is sort of, leading to why are we doing this kind of research is to understand things like this, to understand what makes different human populations or us as modern humans unique and gives us the ability to survive through many of these extreme environments, for example. So I do have quite a few slides on, I wanted to kind of move a little bit because I know some of you may be curious about, the other aspects. So we've looked at, of course, I've given you a snapshot of the archaics. There's still a lot more to it, but I only have, a limited time. But I also wanted to very, very quickly, so I'll maybe take 2-3 minutes if Uma allows. The other sort of part of the highlight was, what do we know about ourselves? So we know now that we have African, sort of origins. We know the timing. We know this contribution from Neanderthals, Denisovans, and different populations. But what have you been able to find out what happened more recently amongst us, all of our population differentiation and similarities? So I just wanted to quickly, sort of throw a few snippets, a few stories. I'm not going to go too much in depth. But just to give you a taste for what sorts of insights we're able to gain. So again, we go back to Africa because that's where, the last 100,000-ish years we have, the modern humans evolving. But these are all sort of examples of different modern human skulls that have been studied from different parts of Africa. And what's been striking is that over at least the last sort of 300,000 to 400,000 years, the range, the morphological range that you find, it's not that you suddenly see, a fossil that is completely modern human. So you do see, even morphologically, these transitioning patterns. So you may see maybe one skull has a certain trait that looks very modern human-like, but you may see another skull that has a different trait that's still modern human-like, right? So there are these sort of heterogeneous patterns of how, what sort of morphological features that we today think about when we think about ourselves. You see them appearing at different sort of times and across different spaces in Africa. So this also sort of means that we cannot really pinpoint that there's this one part of Africa where modern humans just come out of the ground and then start moving out of Africa. This is a huge, complicated mosaic process that we're still scratching the surface in terms of paleoanthropology and genetics. So we expect, even from the genetic, we don't have DNA from any of these because they're very, very old. DNA is not survived. But we expect to think that, maybe our genetic sort of variation and the variability that we see in Africa also sort of formed in patterns here. And so it's been sort of this what we call structure again across Africa. And, whenever we can guess what happened to populations that leave, maybe in the distant future, when we're able to get some sequences, we can understand much better who those ancestors were who sort of formed the path out of Africa. Really, I just wanted to give you 3 very quick case studies of how these sort of patterns, so these sorts of map you'll see in a lot of popular, and I've actually taken this from somewhere on the internet. But a lot of these, again, give you these sort of anchoring dates of when humans reached, out of that Africa migration, different parts of the world. Most of them are based on the first known archaeological evidence in different parts of the world. But I'll quickly give you some stories. The first is what is our understanding of modern European populations? So Europe was, again, you have the out of Africa migration about 40 to 45,000 years ago. You already have presence. So you have modern humans living here. They are all hunter-gatherers at that time. And then archaeologically, they see farming coming in somewhere, because farming originates in the Near East about 12,000 years ago. So archaeologically, you see farming entering Europe about 8,000 years. And then again, archaeologically, people have published that about 5,000 years ago, you see another kind of migration of technologies from this culture called Yamnaya, which is a pastoralist community. So you see this other subsistence coming in the form of pastoralism, in the form of new pots and all of that. So there's at least 3 sort of distinct entities, archaeologically speaking, that have been published. And lo and behold, when you look at the genetic structure of modern Europeans, it absolutely mirrors what archaeologists have said. So genetically speaking, these are all modern Europeans who have been sequenced. Their DNA or their genomes can be actually, in fact, colored as a mosaic of these 3 different ancestry sources. So you have part of their DNA is this blue, which is the hunter-gatherers who lived from time immemorial. And then you have the orange, which is the farmers from the Near East, so their DNA. And this Yamnaya, the steppe culture that comes in 5,000 years ago. So what this is saying in the context of Europe is that both farming and pastoralism come in as people moving in with the technology and with the genes. So it's not ideas that migrate into Europe, but actually new people coming in from other parts of the world. And this Yamnaya culture, in addition to being pastoralist, has also been hypothesized as being, the forebearers or bringing in Indo-European languages into Europe. And this becomes important because I'm going to skip this and move into South Asia because I think a lot of you may be excited to hear about this. There was this big paper that came out a couple of years ago. Most of you must have heard this. And this was the first DNA sequence from Rakhigarhi, which is a Harappan civilization site. And this is about a 4,500-year-old female who was excavated from Rakhigarhi. DNA was sequenced. And this is the breakdown of the Harappan individual's DNA. So most of their ancestry comes from this orange sort of source, which is related to ancient Iranians. And a little part of their ancestry is related to Asian and Andamanese islanders, so the original sort of hunter-gatherer population who are approximated today by the Andamanese islanders. But more importantly, using that DNA from the Rakhigarhi, sort of that lineage, what these authors did was they were able to actually model the ancestry of modern South Asian populations. So they were able to model modern South Asians, much like Europeans, as a product of at least 3 distinct sources. So you have the Andamanese related, which approximates the hunter-gatherers. So you also have the hunter-gatherer component in Europeans. Then you have a second source of ancestry, which is related to the Harappan, sort of, that population. So which again is a product itself of the Andamanese related and the Iranian related. And then most modern populations, all these red dots are all modern South Asian populations. So you see them in this white space, which means that they derive ancestry from all these 3 sources. And the third source is this steppe source. So it's very closely related to that Yamnaya source that goes into Europe, but it's not the same. It's from a similar region, maybe a little more recent in terms of time period. But most Indian populations, if not all, have ancestry from that. But if you remember, the Rakhigarhi individual doesn't. And so the latest model is that possibly the Indo-European languages in India, in Pakistan, in Nepal, and many parts of South Asia, probably also come in from with this migration, much like the mirroring of Europe. You do have some red, I don't know if you can see it at the back, but you do have some of these red dots on this line. So these are all mainly tribal populations from South India, which keep sort of these 2 ancestry sources, but they don't have the steppe ancestry. And these are all Dravidian language speakers, very, very isolated populations in the south. So you do still have populations that haven't been sort of influenced by the steppe ancestry, but most populations, at least the non-northeast populations, have been influenced by all three sources. So even in India or South Asia generally, you see a mirroring of this mosaic patterns of ancestry that contribute to the people living today. And finally, my last sort of story is from the Americas. It's from the work that I did. So the Americas also have been, peopled very recently, at least compared to other parts of Eurasia. You see some of the earliest evidence of humans in the Americas about 14,000 years ago. And people have been debating where people in the Americas came, of course, the ancestors of modern Native Americans. The consensus is that they came over the Bering Strait some sort of 15 to 20,000 years ago from Northeast Asia. Some people also suggest that they came, first sort of settlers came from Polynesian islands into South America and maybe moved up north. And that would explain why some of the earliest dates are in South America. And finally, some people also suggest that people came from Europe through the North Atlantic route and settled here, and later were replaced by migrants from Northeast Asia who wiped out sort of these settlers and became the ancestors of modern Native Americans. And this is based on the similarities of stone tools between the cultures who were living at that time in Europe and some of the stone tools found here. So it has nothing to do with DNA. And in fact, DNA, particularly already from modern DNA, so not even bringing ancient DNA into the play, suggests or highly supports that modern Native American populations are sort of have ancestry that's closely related to modern populations living in Northeast Asia. So that was already established. Yet what we found through the sequencing of a 24,000-year-old little boy that was excavated from a site called Malta in Siberia. So this was part of my PhD project. What we found very quickly here, so this is the grave reconstruction, this is the little piece of bone that I got in the lab. And what we found by sequencing this particular individual's DNA, I'll just show you the model directly, is that this particular lineage is actually what we call ancestral to modern Europeans, people who live in West Eurasia. So it belongs to the same sort of ancestry, but it's a little bit older, so that's why we put them here. But more importantly, they are contributing to modern Native Americans. So the modern Native Americans can be modeled as a mix of East Asians, which was already known based on modern data, but our ancient DNA sequencing of this lineage gave us additional insight that modern Native Americans, almost about a third of their genome, in fact. So quite a substantial part of their ancestry comes from the second source that was harder to tease apart just based on modern sequences. So we needed to plug in this ancient DNA data to be able to suggest a dual genetic origin for modern humans. So far I've given you 3 different case studies: Europe, South Asia, the Americas, and all of them you can see pervasive genetic mixing that leads to the formation of modern populations alive today. So that was really the case I wanted to make. As I wrap up, I'll show you that this is the scene of what we know in terms of ancient DNA from parts of the world. So this is all sort of data sets. The darker, that means more data from ancient DNA from that part of the world. And I've also put in numbers here. These are all data sets in terms of how many genome ancient individuals have been sequenced. You can see the highest numbers are all in Europe. You can see that from the map. This is India for context, only 40 individuals, and sort of contrast that to the population load that South Asia contributes to the world. We know very little about our prehistory based on sort of a lack of ancient DNA data. So I'll do overall takeaways just so we can sort of just wrap up and what I spoke about. So as I mentioned, we have paleoanthropological and genetic evidence that modern humans originated in Africa about 200,000 years ago. Then the sort of event that led to us anatomically modern humans being formed both in and out of Africa is more recent. It's between 60 and 100,000 years ago. That's formed from the recent out of Africa migration. But with the leaky or the introgression model, not the total replacement model, because we do have contributions from Neanderthals and Denisovans, possibly other unknowns, but for now, we at least know Neanderthals and Denisovans. Also, as I showed you through 3 case studies, hope you can appreciate the fact that we're all mosaics in many ways of different ancestral gene pools. And I didn't get into human health and history because of time, but we also have insights on, for example, the hypoxia adaptation that does have a bearing on, of course, our understanding of how different health aspects have evolved over time. I also would be remiss if I could just take, I know Uma's running out of patience with me, but this is my absolute last slide. I do want to say that the kind of work we do is highly sensitive. We are telling stories, narratives about ourselves, about populations, about marginalized communities, about indigenous communities. And so we do have to take accountability of how we portray or how we narrate our science, which is why these sorts of forums are very, very important, because we publish in our jargon, and they may or may not get interpreted in the way that we mean it by the non-scientific audience. And so particularly today, as I mentioned, in light of the underrepresentation, there is a rush for sequencing more and more and more, because that's what scientists do. We want more data. But that also means that we need to keep in mind that we have accountability to local communities, to local researchers, particularly because we're going around minimally, but yet destroying what our biocultural heritage for many, many parts, communities in different parts of the world. We also have to be very, very, careful about conflating or confusing identities. What we build are genetic identities. People have self-identities, oral histories, oral traditions. We cannot compare the DNA data against those. They are completely different identities, yet they get done. So I just want to put it out there. Also, again, public outreach is very important to counter misinformation that's very much out there, particularly in times like this, charged times. Finally, as I mentioned, paying attention to what causes a lot of the underrepresentation, the fact that these are ethically charged, topics, as well as as I mentioned, all of this requires a lot of funding, resources that many, many parts of the world don't have. And so we should be working together to create an ethical framework and work with local communities, local researchers to actually build our narratives in an ethical and a sustainable way. So that's where I'll leave you, and I hope we can get started with the
Thank you, Maanasa. That was an amazing talk. Thank you.
Great. So I have worked a little bit on hypoxia. So I was interested in EPAS1. So EPAS1 is something to do with erythropoietin, is it? It's a transcription factor?
Yeah, it is a transcription factor,
right.
Exactly.
So have you compared, like you did the work on Tibetans, and have you compared it with the Andes where you said?
Yeah, this is not my work personally. There's a lot of colleagues who are working on it. It's at least physiologically, it's a different adaptation. Okay. And so far, the introgression part is only applicable to the Tibetans. So that hasn't been yet detected in the Andean populations. So it's a different phenomenon.
And just to add to that, I think it's very interesting to think also about animals. There are many animals that also live at high altitudes, and there are many other mutations and sometimes no mutations so far identified. So I think that's one of the nice or interesting things about evolution is there may be many solutions to the same problem, and that's why different studies will help us really identify the gamut of possible solutions.
Yeah, you have so many factors, the SOD
yes,
gene.
Exactly. Yeah. Thank you. Sure. Yeah, so while we wait for the next question, Maanasa, it was a really fascinating talk, and as we've discussed, it's something amazing to see the development in this area. You said in the end about potential misconceptions, and this is something which, for example, the study which came out on the Rakhigari individual sparked a lot of debate, a lot of public debate, and kind of comparisons to the Aryan invasion theory and so on. I don't know whether you want to comment on some of those things and how it's nice to have these papers, but how do you? I have a very specific question. How do you communicate this information as found, the evidence as found based on the analysis, when there are existing narratives? So it's similar but different. How do you communicate that nuance? What are your thoughts on that?
I have several thoughts. I think we'll be here all evening, so I'll maybe just share a couple of things here. So first of all, there was recently a study published where they did a really interesting analysis where they looked at sort of different themes, and they looked at who engages with these themes on Twitter, right? And what they found was the sort of, the community, let's say, or communities that tend to engage with the kind of work we do, population genetics, human evolution, they're driven by a lot of extreme nationalist groups. So they often co-opt narratives incorrectly to support, agenda that often are nefarious and maybe hurtful to particularly marginalized indigenous communities, which is why I made it a point to say in my last slide that really the accountability, the onus is on the researchers, because we tend to publish and we think we can get away in a day like today by getting, our jargon-filled papers and washing our hands off it. This may have been possible 10 years ago, but now science is becoming open. It's coming to people's living rooms. My parents didn't talk about mRNA. They don't know what mRNA stands for, but that's the level of scientific conversations that are happening in all our households, and which also then the negative of that is that it does give a huge window of opportunity for misrepresentation, and not all of this is bound to be nefarious. Much of this is just maybe lack of context, lack of proper knowledge about what the results mean, what the data are saying, and so to your specific question, as I said, I think the accountability, the responsibility lies on researchers to now come out along with our papers. Now that our papers are out there, there's so many scientific journalists reporting on this. Again, scientific journalism needs to be tuned up, I think. But in terms of what we can do is we can step out of our labs, into these, particularly these social media where a lot of these conversations are happening online, right? People are becoming very, very vocal. And so, these are sort of the conversations that we need to be part of. We cannot simply say that we've published in our Science, Nature, that we're done, I think the fact that a lot of us are, expending taxpayers' money, our messages should be reaching the taxpayers. And, if there's misinformation out there, particularly if it comes from a space of, ignorance, it can be corrected. Maybe a lot of the agenda-driven conversations, not all of them can be corrected. They have the agenda. A lot of them don't want to listen. But maybe that's also not always your target audience. Our target audience is my parents, is my grandparents, my friends who want to know more, but may not always have the opportunity because they don't have access to researchers. So,
thank you. Any other questions from the audience? Yeah. Yeah, while you're making your way down, I think this is very important. What you say is really important in India today, because there's a lot of consumers of information online and so on. But what information is being put out there may not always be curated.
So my question was, has there been any updates since David Reich and Partha Mazumdar's work in 2016 or 17 or what? I think David Reich and Partha Mazumdar,
yeah.
I think they collaborated with the entire migration theory in India.
He's talking about the
I think
Rakhi Gadi.
the Rakhi Gadi paper, or the series of papers that
Yeah,
came
yeah.
out?
After that, has there been any update? Especially they recently found, I think, a skull in the Kilari dig in Madurai.
Right. So nothing that's been published or peer-reviewed and published. So that's the 2019 series of papers, including the one that I discussed today, was the last sort of ancient DNA-based model that we have. There's more to come very quickly. I mean, I'm working with collaborators here, so I hope there's more models that we can use to refine some of these or, work towards them. But to my knowledge, no, nothing else has been published.
Nothing older than Rakhi Gadi.
No, nothing has been published. There may be older samples that exist,
right,
but nothing that has been put out in the public sphere yet. So there are people working on it, but it's not published.
Yeah, so maybe, that's a great question, and maybe I'll just clarify. One of the things which sometimes happens with science is, like these discoveries of, samples, specimens, remains, and then the problem is the process of science is very long. So, I mean, unfortunately, that's the way it is. So it takes a lot of time to get things into the lab, to make sure that you're doing the work carefully enough. And the process of publication itself, because it's a peer review process, when you write up the work, you submit it for publication, it takes up to a year, sometimes 2 years. So unfortunately, when these things are out, it takes a really long time. And sometimes that time disconnect is something which we have to address. But at the same time, once it's out, it's forever. Any publication is forever, right? It doesn't go away. And so it's really important that we absolutely do the best possible job in representing what we believe is the truth.
One last thing.
Yeah.
I just hope a talk can happen focused on the migration in India at some point. And also, I was very curious to see the slide on lactose intolerant.
Yeah, I skipped through that I can just fill you in. The story there is that, again, lactase sort of persistence, or the ability of adults, human adults, to be drinking milk and digesting is very unique to humans as mammals. All other mammals lose that ability because you wean off your mother's milk in childhood. Why should you retain that? But because milk is such a nutritious source, particularly in times of famine and drought, it's become very integral to our diet over time. Not all populations, East Asians and Native American populations, for example, they don't retain the mutation. So they have the original, the ancestral state, whereas those of us who can, we're actually the freaks. We're the mutants who can still retain and continue to produce the enzyme, the lactase enzyme. So the story that I skipped through is basically this. It's been suggested that this particular this trait, this lactase persistence trait, is one of the strongest selected traits in the history of humans. It's so strong that pretty much, almost 100% of Northern Europeans have retained the ability to drink and digest milk. So it's such a strong pressure that was put, presumably by the need to supplement our diet, that mutation essentially got what we call fixed in these populations very, very rapidly, and as rapidly as potentially the last 4,000 to 5,000 years. And it seems, at least it's been suggested, that part of that, the mutation that came into Europe may have been brought by these steppe pastoralist community, that Yamnaya that I talked about. And then once it entered Europe, again, because until then it was farmers living there, possibly again this new nutritious source came in, and because of the nutrition and because of the fitness levels that it brings to modern humans, then maybe it just shot to high frequency because of the selective pressure. So that's what's been suggested so far. There's been papers that have argued now for and against that, but it doesn't take away from the fact that this particular trait is one of the strongest selected traits of the regions of the genome that we know so far.
Yeah, so just to kind of put that in context, when there's a new mutation like this gene which allows you to, keep lactose, it's going to be very rare. But if people who have it survive better, have more children, which is higher fitness, then over time it quickly becomes common because those individuals are reproducing more. And so what Maanasa is saying is, this is one of the traits or one of the mutations, one of the genes which has been selected the most, which was most advantageous in some sense. So that's really amazing that, there are such examples. And, without being able to look at the past, we can't really infer that. We can try to compare it to populations who don't have this trait or something like that, but it's always conjecture. You're
Just
welcome
one
to
quick
take a
question
look.
with reference to your one slide, where you have shown the linear regression between the distance with reference to Africa and the genetic diversity. So how this genetic diversity is measured? What is the metric for measurement?
So in that particular case, I mean, there's multiple metrics that you can use to measure diversity. So in that particular case, it was heterozygosity. So just in terms of like, when you have different variants at a particular site, so what we call different alleles. So you can measure how many of the different variants are present in a particular population and use that as a proxy for understanding how much diversity exists.
Okay, fine. Thank you.
Thank you.
I have a question.
Of course, please go ahead.
See, is the consciousness of that Neanderthal, that man, is the consciousness of that person, of the Neanderthal, has changed over the years? Have we carrying the same consciousness as that person? Like suppose fear or violence or competition. We are having the same consciousness, or it has changed over the thing?
It's hard to measure those. We don't know that it's got necessarily a strong, or maybe if it does, only genetic basis, right? So a lot of these behavioral traits are very, very difficult to measure in terms of genetic contributions already in present populations. There's a lot of studies, what are called, genome-wide associate, where you sort of measure the trait or the behavior, and you try to see if that particular community or the pool of people have a certain mutation or a bunch of mutations. With behaviors, it's very, very difficult. It can be highly what we call, controlled by multiple genes or multiple variants, but also have an environmental factor on top of that. What kind of household were people brought up in? What sort of early childhood exposure they had? Education, lots of factors. So it's very, very hard at this point to know what sort of conscience these ancient, peoples had. One way to think about it is more from the archaeological perspective. So you can look at, what sort of manifestation, cultural manifestation, material manifestation. So for example, stone tools, cave arts, their burial practices. So a lot of those can give us certainly snippets of, hints about what they may be thinking about symbolism, right? But I don't know that we can sit at this point and do anything but conjecture about the consciousness, and at least definitely from the genetic perspective. So,
Thank you.
Hi.
Hello.
So I had a question regarding DNA sequencing. We've had companies like DeepMind revolutioning the world of DNA sequencing and all of that, and we've recently seen AlphaFold doing all sorts of things. So how do you rather visualize the ethical consequences of those kind of technologies? How do you approach that?
So are you asking specifically like deep mining or just generally just DNA?
Generally, the applications of AI and all of this.
Yeah, absolutely. No, that's definitely a growing concern. Of course, more so for living people, because there can be huge consequences for data piracy or privacy, especially connected with things like insurance, or generally even just the anonymity of the process. I mean, the whole sort of our consent process is based on the fact that we cannot use DNA data to identify a particular person. So we de-identify it right away. But because of the power of the bioinformatics, the analysis, and the power of this genome-wide sequencing, in fact, and these sort of databases, 23andMe, Ancestry.com, where people are readily actually volunteering their genetic information. There have been cases where, for example, perpetrators have been caught in the U.S., for example, because their relatives' DNA was matched with theirs, right, over the years. And so definitely it's a huge issue for people living today. That being said, also from the perspective of ancient DNA, there's a growing concern, particularly again from marginalized or indigenous communities, that many of these migration narratives based on ancient DNA data can be misused to, let's say, have negative consequences for things like their national identities or their land rights. So I think we definitely need to take that into consideration. There are already conversations happening in the research arena. How can we better safeguard data? We are just sort of stakeholders here. We are caretakers. We are not the owners of these data. And so when you put it out in the public sphere, what can go wrong? We didn't think that ancient DNA data could be misused, but it's possible now that we have seen it being misused, that it's possible. So there are ways that you can safeguard. For example, you can set up data access agreements. We still need to make it public because things need to be replicated. That's a scientific process. But you can still control how that, beyond the replication, how the data can be used or, let's say, misused. So there are definitely things that are being talked about, but it's a huge concern. So thank you for bringing that up.